Patterson here. Today we're going to talk about passive transport across a membrane. This is part of AP Biology Unit 2. When we look at a plasma membrane, remember I have a phospholipid bilayer. These are my hydrophilic heads and my hydrophobic tails. This area loves water. This area does not. So you have different types of properties with this plasma membrane. Remember, a plasma membrane is one of the three items that all cells have. All cells have a plasma membrane. The other two is going to be DNA and cytosol, but they're all going to have some type of plasma membrane. Now, they can be a little different, but all of them are going to basically have the same type of function. They all might look different, but they all have the same type of function. So they can regulate. They're going to allow material in and out. They're going to be very selective. It's going to support and protect. It's going to allow a framework for the cell to be able to do what it does. It's going to protect it from things outside the environment and not allow it in. So it's going to help protect. It's going to be a barrier. It's also going to be very flexible, so it can move and shift and change its shape a little bit if it needs to get past something. So this is a very fluid model. Remember, our fluid mosaic model means that I have pieces in it and that fluid, it moves. This whole membrane can move. So this protein can come over here, it could go here. This carbohydrate chain, this glycoprotein can come over here. All right, so all of these things can shift and move. A membrane is also what we call selectively permeable. It means it's going to be able to let certain material through, and it can keep or impede other material from coming through. So here I see my hydrophilic head, my hydrophobic tails. This is going to allow material or molecules that are small, nonpolar, or gases to go through. So here I have hydrophobic. They're going to want to go through. They're going to be attracted to these hydrophobic tails, and they can just jump right through this plasma membrane. I have carbon dioxide. It's a gas. It's going to go through that membrane very easily. Here I have oxygen gas. It's going to come through here. And this one kind of stumps people. You have oxygen, remember, very electron negative, very greedy for electrons. How is it able to just go through? Well, this becomes nonpolar when it bonds with itself. So oxygen gas is O2, and since oxygen bonds with itself, it becomes nonpolar. And so it's able to come through, and it's also a gas. I have selective permeability. I have large, charged, and hydrophilic. They're not going to be able to go through this membrane on their own. So they have to have a pathway or a passage that's going to allow them to get through this. Some of them can be gated. Some of them are going to have like a charge that's going to allow them to come through. But there's going to be something very special about this. So here I have transport proteins, and here I have aquaporins. So some of them will be very specific. So aquaporins is going to be water. Transport protein can transport things like sugar or things that are charged. So here I have sugar molecules. So these sugar molecules will be attracted to this transport protein, and then I'll have sugar molecules that will go through. Here I have aquaporins. This is going to be charged where it's going to attract water molecules, and those water molecules will go through too. I have what's called a concentration gradient, and it's a difference in the concentration amounts on either side of the membrane. So here you can see I have just a few pieces of substance. I have a few molecules over here, but over here I have a bunch. These molecules do not want to naturally come here. They want to go to a place where they're more spread out. So this kind of works with people too. When you have a room full of people, they're not all bunched together. They spread out, and they're going to go to a place where there's not a lot of people. And so this is kind of what molecules do too. So I have a bunch over here, and they want to spread out. So they're going to naturally go through this membrane and come to the other side. So my low concentration, my high concentration, they're attracted to the low, and so they're going to move through the membrane and go to the side that's low. I also have what's called an electrochemical gradient. This is the difference in concentration charges on either side of the membrane. So let's look at sodium. Sodium is this one right here. Okay, I have 1, 2, 3, 4, 5, 6, and over here I have 1. This sodium is going to be attracted to the sodium charge over here, and then they're going to come through this gradient. I have potassium. I have all these potassiums over here. I have 1 potassium here. They're going to naturally go through this. We're going to talk about some later where it's going to be attracted to the negative or the positive charge on the other side. But for right now, this is kind of what you need to think. But I've got more potassiums over here, so it wants to go this way. So my potassium gradient goes this way, sodium goes this way. Here I have active transport, and active transport, I need energy, and that's going to require ATP. And ATP is the energy currency of the cell. Whenever I need energy, ATP is going to come in, and it's going to help give it just that extra boost to be able to move over this energy hump to be able to allow whatever it is to have the energy needed to do it. So here I have molecules. I have 1, 2, 3, I have 5 molecules here, and I have all these other ones here. There are some processes inside the cell where it needs a huge buildup of molecules on one side. So ATP has to keep forcing these molecules to go. Remember, they don't want to go here. They want to go through this membrane where it's lower. So this ATP is forcing these molecules to be here. And this usually gets used later on. We see this in cellular respiration and photosynthesis. So I have a low concentration, high concentration. It's going to force it here. And we're not going to really get into that right now. So we're going to do this video a little bit later. So active transport will be in a separate video. So let's go back to passive transport. So with passive transport, we don't need energy to do that. They're going to diffuse, or they're going to go down their concentration gradient. Here I have a few green dots. I've got a lot over here. So these dots are going to want to go where I have lower amount of green dots. Same thing with these brown dots. They're wanting to go where I have less brown dots. So low concentration, high concentration. So I'm going to move towards low concentration, or they say this is going down my concentration gradient. I have different types of passive transport. I have facilitated. Facilitated means I have a helper. So if you ever have a facilitator in a classroom, that is somebody that's in there helping the teacher or helping the aide or helping a student. There's something being facilitated. So the transport proteins is facilitating diffusion. It's helping to diffuse these items in. Because remember, things that are large and charged and large, charged, and hydrophilic, these are not going to want to go through this membrane. So I have to find another pathway for it to go. I have osmosis. Osmosis just deals with water, and that's going to be wanting to go through something, and we have aquaporins. Water technically can go through these membranes. It doesn't like to. It takes forever for it to get through and forever for it to come out. So these aquaporins move really quick. I can move just billions of these every second. Diffusion is just going to be matter. Remember, gases, small, nonpolar, are going to be able to move through this membrane very quickly. So with diffusion, I have hydrophobic, I can have gases, I can have nonpolar, and they're going to be a constant movement through this membrane. So this is going to happen spontaneously. There's going to be nothing that's really going to get it started except for the concentration of molecules on the other side of that membrane. They're going to move down that concentration gradient, and they can diffuse at different rates. Okay, so I can have some that move real fast, I can have some that move kind of fast, and some that move really slow. So they can diffuse through at different rates. So with osmosis, we talk about just water. So diffusion of water down its concentration gradient. Remember, I'm going from a high concentration of water to a low concentration of water. Here I have membrane, and here I have aquaporins. So these water molecules are going to want to go down through these aquaporins from a high concentration of water to a low. So water follows high concentration of solutes. I tell my students that water follows the solutes. So this is what that means. Here I have 20% glucose in a solution. If I have 20% glucose in the solution, that means I have 80% water. Here I have a dialysis bag. I have 5% glucose. So in this solution, in this bag, I have 95% water. I have more water molecules here, so I have a higher concentration of water. I have a lower concentration of 80% water out here. This water inside this bag is going to want to naturally diffuse through osmosis through this bag. But when we talk about solutions, we're talking about usually the solute, what it's made of. We don't really talk about how much water's in it. So when we talk about solutions, I tell my students, wherever I have more solute in a solution, most of the time the water's going to go in that direction. So again, I have 95% water, I have 80% water, I have more solutes in here. So what that means is these solutes are taking space of spots where water could be. And now I have more solute taking up space for water. I have less solute taking up space for water. And so this is why I tell my students, water is going to follow the solute that's the largest, because that means I have less water because I have more solute there. It's kind of a backwards idea, but once you kind of get your head around that part of it, it helps out. But again, I have a high concentration of water to a low concentration of water, and this is a lower concentration because I have more solute taking up space. Facilitated diffusion, no energy is needed. It's going to move through transport proteins. So again, these are proteins that are embedded in that plasma membrane. So my membrane here, my phospholipid bilayer, these proteins are inside, and they're going to move through again from a high concentration to a low concentration. So here I have a few dots. It's going to want to move this way. I have a few green dots. It's going to want to move this way too. So from a low concentration, that's where the solutes down here, or the molecules, are going to want to go. I have channel proteins. This is going to be a pathway to allow molecules or ions to pass. And here I have channel proteins here. Some can be charged. So if I have charged ions coming through, I could have a charged protein that's going to attract this specific type of ion through. I can have other ones that are going to attract certain types of polar molecules, so like sugar or some type of charged molecule. So these proteins are going to be kind of very specific to what type of molecule it wants to come through. Aquaporins are going to attract water, and this is going to be a pathway. These are all proteins that are going to be made to be able to allow these molecules through. They're going to help with hydrophilic, so these are going to be hydrophobic on the outside, but they can have a hydrophilic center. So they're going to be very specifically made to be able to live inside this embedded plasma membrane. They're going to be gated. It's going to allow movement when there's a stimulus. So sometimes this may not be charged and then it can charge itself up. I might have some type of gradient that's going to have to be created before it works, but there's going to be some type of stimulus that's going to allow it through. I have carrier proteins. These are proteins that are undergoing a conformational change. So this is going to be a molecule that comes in. When the molecule comes in, remember this is a protein, okay? The molecule comes in, it's going to attach to this protein. And I'm going to change the shape of the protein. If I change the shape of the protein, I change the function. So now it attaches, it's going to close. When it opens back up, this molecule is going to come out, and then when it comes out, it's going to cause another conformational change and cause it to come back here. So the molecule comes in, it's going to mess with the protein, cause it to change, it's going to close, open up at the bottom, it's going to release, it's going to now make this protein go back to the way it originally was, and then it's going to allow another molecule to come through. So this will be some type of conformational change because of the protein. Remember, you change the shape of a protein, you change the function. Okay, That's it for passive transport across a plasma membrane. I hope that was helpful. If it was, I'd appreciate if you guys would subscribe. Let me know if you have any questions. We'll talk to you guys later.